Hello and welcome to New Scientist Weekly, home of the most fascinating science and technology news. I'm Christy Taylor in New York.
And I'm Timothy Revel in London. This week on the podcast, we meet a transparent mouse. The rodent, that is, not the device. We'll talk about why the US military's latest high-tech spy gear is a balloon, and the seemingly inescapable relationship between human health and bats.
And how dark is space, really? You'll have to listen and find out. But first, we often hear overhyped news stories promising new cancer treatments or even the C word, cures. But they also come with big caveats. But this week, our reporter in Australia, James Woodford, has been covering what really does seem to be a very significant breakthrough that may have major implications for the treatment of blood cancers, namely the first successful creation of human blood stem cells in the lab. Hey there, James.
G'day, Christy. Thanks for inviting me in to discuss this.
Yeah, absolutely. It's a super cool sounding story. I gave the top line already, but what exactly has this research team achieved?
Yeah, right. The top line alone, that human blood stem cells have been grown in the lab, is itself pretty amazing. But to go just a little bit more in depth, the scientists from Murdoch Children's Research Institute in Melbourne have achieved a two-stage process for creating these stem cells in the lab. And the conclusion of that process is that when these cells are injected into immune-suppressed mice, the stem cells successfully become bone marrow and blood cells.
I mean, that sounds amazing. And I guess what I want you to do is take me back to why this would be a big deal. How could it change cancer treatment?
Yes, specifically, we'd be talking about blood cancers like leukaemia and lymphoma, which we treat with radiation and chemotherapy. Now, the downside of these therapies is that they can destroy the blood-forming cells in bone marrow, so they need to be replaced by new, healthy ones. The best way we've currently got for that is umbilical cord stem cells, which come from a very limited pool of donors. And since it's tissue from someone else's body, cancer patients' bodies may reject the new bone marrow. So scientists have been really wanting to find a way to use patients' own stem cells to replace their own bone marrow. And that's what this new research seems to offer.
Yeah, and you mentioned a two-step process for making these cells. At the risk of getting maybe just a bit too technical, what were those steps and how did they manage it?
First, as Andrew Elefante, one of the researchers, explained to me, they use a process called reprogramming to turn human blood or skin cells into so-called pluripotent stem cells, meaning that they can now become any type of cell. And this involves temporarily turning on 4 genes, which allow the original cells to revert to an earlier stage of development where they haven't yet become a specific type. And the second stage involved turning those pluripotent cells into blood stem cells. They first make thousands of small floating balls of cells, a few hundred cells in each ball, and direct them to change from being stem cells to sequentially become blood vessels and then blood cells. This process, called differentiation, takes about 2 weeks and makes millions of blood cells, Elefante says.
Yeah, so very cool, kind of harnessing what stem cells naturally do in the body in the first place. So this is the first time that we've coaxed human stem cells to become blood stem cells in the lab. How well did it work on these mice that you were talking about? And just to be clear, we are talking about human blood cells implanted in mice?
Exactly, yes. Human blood cells in mice. So when they implanted these human cells in mice whose immune systems had been knocked out, the mice did go on to develop functioning bone marrow about 50% of the time. That means their bone marrow was making the cells that carry oxygen and fight infections, all the key jobs that our blood does. And this success rate is comparable to what we see in umbilical cord blood transplants.
And as you said, if we can use cells from cancer patients themselves to create these stem cells, it sounds like we'd reduce the risk of rejection. Is there any kind of risk, though, to making blood stem cells from someone who already has a blood cancer, though? It sounds like it could come with, some contamination risks, for example.
Actually, I found this part of the story really surprising, and also surprising how confident that the researchers would be that they'd be able to deal with this issue. The researchers told me it's now pretty straightforward to edit the genes in the lab-grown stem cells to remove any DNA that could lead to disease again.
That's amazing. So we've taken human blood stem cells and proved they can, at least 50% of the time, lead to functioning bone marrow and blood in mice. So when do humans benefit?
There's no guarantee that what's worked in mice will be effective in humans, and there's a long way to go before clinical trials in people can begin. The scientists say that this is a very important first step towards new treatments, and the team hopes to see testing on people within about 5 years.
Now, for a look at something else, or should I say a look through something else. We've got an incredible story about how researchers have managed to make living mice transparent. They have literally turned a normally opaque animal see-through, so you can see its organs and just generally its innards. There are some really quite graphic pictures and videos of those that made my stomach turn, but the animals are, surprisingly, they're doing just fine. I guess we're just not used to seeing the insides of a rodent, especially a living one. To tell us how this one works and if we'll be able to turn ourselves transparent anytime soon, we've got assistant news editor Chris Sims. Hi Chris, welcome back to the show.
Hello.
So, transparent skin. How did they work this one out?
It's actually deceptively simple. If you want to see through skin, you simply rub a specific but very ordinary food dye onto the skin that you want to peer through. With ordinary skin, you could think of it as trying to peer through fizzy water that's full of bubbles because you've just shaken the bottle. Water and gas have different refractive indices, so light shining through this gassy fluid changes direction every time it goes from water to a gas bubble or from gas to water. This means light scatters in all directions and can't penetrate through the water as easily as it can through just air or clear water. You can't see through it. Skin, like other biological tissue, behaves similarly because we contain a lot of water, but also other molecules like lipids and proteins that usually have higher refractive index than the water.
Okay, well, I mean, that sounds quite complicated, yet it's just food dye that's applied to the skin. Can that just change that? Is it that simple?
Yes, it is. Well, the food dye they used, which is called tartrazine or E102 or yellow 5, changes the refractive index of the water inside your tissue to more closely match that of the lipids and the other molecules. This reduces how much the light scatters. That means you can see further through it. Rather than just seeing a millimetre into a body, once you get your microscope out, you might be able to see a centimetre. For example, massaging the dye onto the skin of a mouse's belly allowed researchers to watch blood vessels and organs at work. They could see peristalsis, which are the muscle contractions that move food through the digestive tract, as it happened, and they could look into the blood vessels on the surface of the brain. And perhaps best of all, the effect is temporary and doesn't seem to harm the animals at all. Once the dye is washed off, the mouse returns to its normal, solidly visible state.
I find it so amazing that this is just a food dye we've always had, and now we know it can turn skin transparent. And I do have to ask, could you do this with humans? Have the researchers tried, even just like on their thumb by accident while massaging a mouse's belly with this dye?
Well, the technique hasn't been tested yet on humans officially, although it also worked on slices of chicken breasts and would absolutely work in us too. But even if all the soft tissue in your body was stained with food dye, this wouldn't be a Harry Potter invisibility cloak. Your skin would probably temporarily have a bit of a glassy look in normal light. And even if the effect happened through your entire body, you'd still be seeing bones and the organelles inside cells. Overall, the effect's a bit gruesome, if I'm honest.
Yeah, I can imagine it's rather gruesome, though I personally am still very curious to see peristalsis in action. But I'm presuming there's also some serious scientific use for this too. It's not just a cool mouse party trick.
Yes, absolutely. The potential is huge. The jump in how far you can see is an increase of a factor of 10, while most advances in microscopy give you a few extra percent. So the technique could help doctors look deeper into our bodies to diagnose issues or help us probe the workings of living individual cells. It could also let us see further into living, functioning brains than ever before, which would be a boon for mapping out which parts do what, something we're still learning about only slowly.
Okay, and we'll put those gruesome photos on our website at newscientist.com if you're brave enough to have a look.
In early 2023, a Chinese balloon caused an international kerfuffle after drifting across North America. Over the course of a week, it drifted eastward across Alaska, Canada, and the contiguous US, while China claimed it was merely a misplaced weather balloon. That balloon was later shot down by US fighter jets and found to be full of intelligence-gathering equipment. Now, the US military is busily planning to launch its own high-flying balloons capable of either performing surveillance or acting as backup communication links. Tech reporter Jeremy Su is back to talk with us about the apparent resurgence of spy balloons. Hey, Jeremy.
Hello again.
So I want to start out by being clear on what kinds of balloons we're talking about. These are not just your average party balloon. They're actually bigger, fancier, heavier than your typical helium or mylar situation, right?
Yeah, that's right. These balloons are often as tall as multi-story buildings, and they can carry equipment loads about the size of a regional jet slung beneath them. They also have solar panels that are often attached to keep all that supplied with electric power, and they're capable of flying at altitudes of 18 kilometers or higher into Earth's stratosphere while using computer-powered weather predictions to ride the winds to wherever they want to go.
And as we've kind of alluded to, these are useful for either surveillance or as something like a backup communication link. I do have to ask, though, why balloons? What advantage do high-altitude balloons have over aircraft, drones, satellites, all that other fancy, newfangled stuff in performing military missions and remaining undetected?
One big advantage is that these balloons are relatively cheaper and more disposable than all the usual options, so that means you can also potentially send up a lot more of them. And as the 2023 spy balloon saga also showed, the balloons are actually often hard to track by radar, and so if you want to get rid of them, you may also have to use expensive air-to-air missiles.
So conveniently, we're talking about this, about a year after that saga with the previous spy balloon floating over the US. Is this just a case of the US now thinking, okay, that seemed quite effective, we'd like some of these ourselves, or has the US been looking at them for a bit longer?
Yeah, the US military has actually been experimenting with these high-altitude balloons for years. So on the one hand, we can't really say that the 2023 incident suddenly opened US eyes to the possibilities there. But on the other hand, it's possible the incident spurred an acceleration of U.S. efforts. Recently, the news publication Breaking Defense reported that the U.S. Department of Defense is fast-tracking development and deployment of these balloons through a special program that's meant to bypass all that bureaucratic red tape. I also spotted several recent U.S. government notices that describe U.S. Army efforts to develop radar or laser-based 3D mapping sensors that can operate aboard these high-flying balloons.
So do we know where these military balloons might end up being deployed around the world? What are the plans for them?
It's most likely that most of the balloon action will focus on the Pacific as the U.S. and China continue building up their respective militaries. The U.S. Army has been launching balloons during military exercises in Guam and the Philippines this past summer. And at the very start of 2024, China actually launched more than 100 balloons that flew either near or over the island of Taiwan during the Taiwanese presidential election.
That's a lot of balloons. And given China's relationship with Taiwan, I'm guessing they weren't meant to celebrate the Taiwanese elections.
Right. Most likely these balloons were perhaps intended as a warning or intimidation tactic that was aimed at Taiwan's self-governing democracy. China claims Taiwan as part of its territory and has consistently deployed military aircraft and ships near the island.
Now, bats have long played a role in human health, as famously they sometimes harbour disease that can infect us too. Just look no further than the virus that caused COVID-19, which came from a bat-borne virus and then, probably via another species, reached us. And there's also their role famously as reservoirs for the rabies virus. But what happens when bat populations collapse? New research this week has found that too affects human health, but counterintuitively, also in a bad way. Reporter Michael LePage wrote about a study this week of how both insecticide use and infant mortality are going up in parts of the United States after a deadly fungus wiped out local bat populations. Hi, Michael. Thanks for joining us.
Hello.
So talk us through this one. What did the study actually find? How dramatic were the effects of bat population decline?
Oh, it turns out they are pretty big. So bats eat a huge number of insects, including many pests. Once this fungal disease arrives and starts killing bats, insecticide use on farms is going up by 30% on average. And even though farmers are using more insecticides, they still seem to be losing more of their crops. So the money that farmers earn from selling crops is going down 30% too. And the thing is, this disease, which is called white nose syndrome, it only affects 11 of the 50 or so bat species in the US. So this is just what happens when you lose some bats, not even all of them. Now, the other thing that the study found is that the number of children who die before reaching 1 year of age went up by 8% too in the regions where this disease has arrived. Now, that figure translates into around 1,300 extra infant deaths by 2017, which is how far the study goes up to. And of course, since then, white nose syndrome has kept on spreading, and so the true number of infant deaths could be much higher now.
Okay, but I mean, that makes sense that when the bat populations collapse, more pesticides are needed. But how does that link to human health? Why are more children dying?
So the author of the study, E.L. Frank at the University of Chicago, puts it down to the rise in insecticide use. Now, because the rise in infant death is happening locally in the same places where the insecticides are being used, this could be a result, say, of people breathing in these insecticides when they're being sprayed on fields, or it could be because water supplies are getting contaminated, but we don't know exactly. The study doesn't tell us that.
Are we sure this isn't just a correlation, though, Michael? I mean, how can we know that infant mortality went up because of the loss of bats and not some other non-bat reason?
Yeah, that's a crucial point. So usually the best that studies can do is just to show a correlation. For instance, you might find that places with fewer bats use more insecticides and have more infant deaths, but that doesn't prove cause. But what we've got with the spread of this white nose syndrome, it's kind of like a natural experiment where you get rid of a lot of bats and see what happens. And what we've seen is that whenever this white nose syndrome arrives in a region, insecticide use starts to go up, and then so does the rate of infant deaths. So Frank thinks that his study demonstrates causality, not just correlation. He thinks he's shown that the loss of bats really is a cause both of more insecticide use and of more infant deaths.
I mean, I do have to say 1,300 more children dying because of higher insecticide use sounds really bad. And it does make me wonder if there's a bigger problem than the bats. Like, maybe insecticides are more dangerous than we give them credit for. Do we need to be doing more to curb their use?
Oh, absolutely. We should be doing all we can to minimize the use of all pesticides. There's no doubt about that. But when I asked another researcher who studies the effects of pesticides about this bat study, he pointed out that the loss of income would also lead to more stress in farming communities, and it's also been shown that this kind of stress can in itself lead to more child deaths. So it's possible that the rise in infant deaths isn't entirely due to higher insecticide use alone. That doesn't mean it's not been caused by the loss of bats, just that there might be other factors besides insecticides at play.
It sounds like even with all of those factors, bats are playing a much more complicated and important role than maybe we realised.
Yes, I think nowadays we talk a lot about the importance of wildlife and biodiversity, but it's in these sort of big abstract terms. This is a great concrete example of why it matters. So you take away key insect predators like bats, and then farmers have to use a whole lot more insecticides. And even when they do that, they've still got more crop damage, so they're still losing money. And those losses, by the way, mean that we're all going to end up paying more for food because it's not just the farmers who are affected. And on top of that, we've got these health effects in terms of more infants dying too. That's a really dramatic demonstration of why biodiversity is something we all need to care about and do a whole lot more to protect. Because right now, we're not doing nearly enough and biodiversity is still plummeting in most places.
All right, Tim, one last couple of stories from me. First of all, I've got really into riddles lately, and I have one for you now. How dark is space?
Okay, really, really dark. You're setting me up here. There's some sort of trick for sure.
Well, there's no fun riddle-like answer to this, but the big thing to keep in mind about space is that there's never anywhere that there is zero light. Even when you get as far as you can from any particular galaxy, there's going to be a very dim glow from the combined light of all the galaxies in the universe, which is a lot of galaxies, even if they're all very far apart. So we can't say that there are zero photons pinging around in the most remote regions of the universe. But measuring that background light from our position on Earth is very tricky. We have all this interference from all of our very bright nearby light sources. So enter the New Horizons spacecraft, which NASA initially sent to look at Pluto and is now sort of plugging its way to the edge of the solar system like the Voyagers.
Okay, and so it's shining light on darkness. I see what I did there. So go on, how dim is that glow then, Christy?
Right. So I'm going to let the researcher on this paper say it for us because he has the best image. He is a fellow named Mark Postman, and he says that it's as if you were in a deserted cabin on a moonless night, more than a kilometer from your nearest neighbor. So picture that. Mm-hmm. Now, if the neighbor opens their refrigerator and that little light in there turns on, again, a kilometer away from your cabin, there will be a very, very tiny change to the amount of light on the wall of your cabin. That is the dim glow of the universe, and it's still 100 times darker than the darkest sky we can see on Earth.
That is really awesome. It's really hard to imagine what that would actually be like, a fridge light from a kilometer away. It's also a little bit scary just imagining it being that dark, especially with a deserted cabin thrown into the mix.
Yeah, just floating around in space, right? Well, if you, like me, feel that spooky season properly begins in September, imagine being on the International Space Station, that much closer to the darkness of space. And maybe there's some weird noises coming from the Boeing Starliner capsule that got you stuck there.
Oh, yeah, I heard about this one. Those two astronauts that took Boeing's capsule up to the ISS for a short stay of just a week and have now been stuck there for months. It's those two you're talking about?
Yes, those astronauts. They're Butch Wilmore and Suni Williams. Now, Wilmore recently reported some odd pulsing pinging sounds, kind of like sonar, coming from a speaker in the Starliner capsule, which to me sounds like the perfect setup for a horror movie. Stranded in space, you don't know how long, weird noises.
Yeah. I mean, it does. And in space, I imagine any strange sounds seem a whole lot stranger. I know there was a lot of interest in this particular story. Did NASA manage to work out what the strange sounds were?
Yeah, and unfortunately, the real answer is pretty boring. I mean, unfortunately for me, fortunately for all the astronauts involved. Basically, the ISS has a very complicated audio system, and sometimes new visiting craft create interference and feedback. So NASA has also now said the sound has stopped. Basically, they figured it out. They sent in their best podcast producer. Don't worry about it.
Don't worry about it until it's too late.
Spooky season.
All right, that's it for this week. If you like this show and think more people should know about us, please do give us a 5-star rating or even a review on Spotify and Apple Podcasts.
We've got more fascinating stories from New Scientist on our website at newscientist.com. Please ignore the suspicious pinging sound. We'll be back next week. Bye for now.
Bye.
This podcast is produced by OG Podcasts. Find out more at ogpodcasts.co.uk.